of Producers Club events. As you know, we've been doing these over the last few months as we've all been sheltering in. And today I think we've got what I think is one of our more exciting events that are coming up, because it's very topical in what's happening in our world. We'd like to thank the Jolla Institute of Immunology for offering us an exclusive opportunity to hear more efforts that are being made to create an effective COVID-19 vaccine. And joining us are Dr. Shane Crotty and Dr. Alex Sette, and we thank you both for joining us. These are very talented researchers and scientists, and we're just not going to jump right in, because we all know how COVID-19 has been affecting our city, our county, our country, our world, and how important a vaccine is. So let me just do a quick intro for them, and I'm going to turn it over to both of them. Dr. Crotty and his team study immunity against infectious diseases. They investigate how the immune system remembers infections and vaccines. By remembering infections and vaccines, the body is protected from becoming infected in the future. Vaccines are one of the most cost-effective medical treatments in the modern civilization and are responsible for saving millions of lives. Yet good vaccines are very difficult to design, and very few new vaccines have been made in the past 10 years. A better understanding of immune memory will facilitate the ability to make new vaccines. Dr. Crotty's lab is hard at work on candidate HIV vaccines with the Shavid Consortium. His lab is also hard at work on vaccine strategies for influenza, strep throat, and COVID-19. And also Dr. Sette has devoted more than 35 years of study towards understanding the immune response, measuring immune activity, and developing disease intervention strategies against cancer, autoimmunity, allergy, and infectious diseases. Dr. Sette's lab approach uses specific probes to define the immune signatures associated with protective immunity versus deficient immunity. I'm sure you all understand that. This research will improve our understanding of how the body successfully battles infection, and conversely, how pathogens escape the immune system, causing the individual to succumb to disease. And so what I'd like to do now is turn it over to both Dr. Crotty and Dr. Sette for their presentations. And thank you both very much for joining us today.
Thank you for having us.
Yeah, happy to do it. So I'm going to lead off, and then Alex will talk, and we will switch back and forth a little bit. He and I have been collaborating so much we can finish each other's sentences. So I definitely expect this to go smoothly. So this is the two of us. I actually spoke at a KPBS event last year, and I actually spoke on pandemics and vaccines. And one of the things I said was that pandemics will happen again. And in fact, unfortunately now that's what we're facing. And so, as was said, Alex and I are going to talk you through some progress towards COVID-19 vaccines and what has to be understood to better control COVID-19. And we focus on the immune system and vaccines. So a little bit of an intro on vaccines is that they are incredibly effective. This is one of my favorite graphs, which is of measles, and this is all 50 states in cases of measles during the 20th century. And then this is when the measles vaccine was invented and introduced, and it's completely wiped out measles in the United States and in much of the world. And measles is actually quite a nasty infection. And so most people don't know that the measles vaccine has actually saved over 14 million lives in the past 10 years. And people don't notice that because those are people who never got sick in the first place, right? That's the brilliance of vaccines. They keep you from ever getting sick. They keep the virus from spreading. They can work incredibly well. And there was a poll question about how many vaccines are there, and here's actually a list of all the human vaccines and when they were invented. And it's a nice list, but we'd love for it to be a substantially longer list. What my lab focuses a lot on in a regular year is how the immune system functions as it relates to vaccine development, to try and make vaccine development easier and faster by understanding how the immune system responds to vaccines. And that's because vaccines completely depend on the immune system. Okay, vaccines only exist because of immunological memory. So a previous encounter with a pathogen teaches with a virus, let's just say, let's focus on viruses today, with a virus teaches the immune system to recognize and remember that exact pathogen. And that's called the adaptive immune system, okay? And the adaptive immune system has two jobs: recognize something and then remember it. And so the adaptive immune system is the part of your immune system that say recognizes polio as specifically being polio and can tell the difference between that and COVID-19. And has various tools to protect you against those in very specific ways, right? So the way the immune system responds to protect you against polio is only good for protecting you against polio. It's very specific, and that's fantastic. That's brilliant. But the problem with the adaptive immune system is that it's relatively slow. It takes time to recognize a new infection. Okay. And so the brilliance of vaccines is to take time out of the picture. The whole point of a vaccine is to expose you and teach you what that virus looks like before you ever see the infection, so that when you do end up seeing the virus, you've already remembered it, basically, and you already have an immune response that can protect you against that. And one general question about vaccines is since they depend on immune system memory, immunological memory, well, how long can a vaccine work for? And one proxy for that is, well, how long does human immunological memory last? And I actually did experiments to answer that, studying the smallpox vaccine, which was a really perfect example for that, because smallpox had been eradicated and most people didn't get vaccinated anymore. And there were no viruses circulating. So anybody who had immune system cells that could recognize smallpox had those cells from an exposure to the vaccine years before. And so when we looked at people who had been vaccinated 30, 40, 50 years ago, I could find each dot as a different person. And if I did the experiment on young people who hadn't been vaccinated, I detected no immune cells in their blood that could recognize smallpox. So that made sense. But all these people who had been vaccinated 30, 40, 50, 60 years in the past still had memory cells circulating in their blood that could recognize smallpox from that one vaccination they'd gotten in childhood, 50 years ago. And so there are cases where immunological memory can be very impressive and long lasting, but not all immunological memory is that way. Some immunological memory is short-lived, and that's not well understood, and it is something important to understand. But we at least know it's possible for vaccines to elicit immune memory that can last your lifetime. Okay, so that brings us to a couple of key aspects about vaccine development for COVID-19. Okay, and so when one tries to develop a vaccine, one has to keep the immune system in mind, and several different aspects of the immune system are important. And one key feature is usually almost all of the vaccines that currently exist for diseases that people can normally cure themselves of. That is to say, an average case of polio, a person survives, and they survive because their immune system responded, recognized, and cured them of that. And so COVID-19 is an acute infection that resolves or cures in most people. And so that suggested that the immune system probably makes a successful immune response to COVID-19 in most cases. But that was unknown when Alex Sette and I started these experiments, and he's going to talk you through that. But that's an important parameter. And questions we then had were, well, how much of an adaptive immune response is there to COVID-19? That's important because if people normally don't respond to COVID-19 or respond to the wrong parts compared to what people are trying to make a vaccine against, that would be a red flag for vaccine development. And so therefore, it's very important to study people who have actually had COVID-19 disease to understand how to best make a vaccine against COVID-19, okay? And that connects to memory as well. And here on this final bullet point is really the most important bullet point, is that your immune system is not one thing, okay? Your immune system is a very complex place, lots of different cells, lots of different jobs, lots of different functions. I wish the immune system was a really simple place because it would make my job a lot easier, but it's actually a very complicated place with lots of moving parts, lots of different cells. But at its core, there are three parts of the immune system that are important for protection against infections like COVID-19 and are therefore important for vaccine design. So one part is antibodies. And so most vaccines are based on antibody responses. And so we want to know, do people make antibody responses against COVID-19, and what are those antibody responses like? But that's just one of the three parts. And the other two parts are actually called helper T cells, or CD4 T cells, and killer T cells, or CD8 T cells, okay? Antibodies are great because they stop viruses from infecting cells, okay? But once the virus has infected the cells, the antibodies usually can't do anything. And so the immune system has a different cell type for that job, which are the killer T cells. So their job is entirely about finding infected cells and killing those cells. And so these are complementary jobs, right? And then in the middle are the helper T cells, which actually help both cell types. So you can't get a good antibody response unless you have these cells. And these cells have independent functions that are antiviral. So in fact, for original SARS, SARS-1, people found in animal studies and mouse studies that actually these cells of the immune system could completely protect against SARS without these or these, okay? And so for each type of viral infection, it's important to know which parts of the immune system actually respond and which of those are actually protective, all right? And so with that as a starting point, that's where we were when we started our COVID-19 studies, which Alex will take over from here.
Thank you, Shane. So this slide, in a cartoon fashion, highlights what has been our approach to the research of COVID. So the virus is it's a relatively complex thing. It's made of 25 different proteins, and each protein is made of many different pieces. And our approach has been to basically take the virus apart systematically and make, in the lab, synthesize all the different pieces that correspond to the whole virus. Then, once we had these pieces in hand, to go in the lab and put the pieces together with blood draws, with lymphocytes derived from various different people, and see what lights up. What does the immune system recognize in this in the virus? Which pieces of the virus are recognized by the immune system? So in the next slide, or series of slides actually, I'm just going to guide you through a kind of a roller coaster has been the last few months, and started out in late January. One of the things that also my lab does is runs this immune epitope database where we catalog constantly all the immune reactivity that is described in the world. And we run this for the NIH. It's a freely available resources to everybody on the planet, all the scientists and all the vaccine developers. And so in late January, come as a blip, this results that this news that in China there was this new virus, and Bjorn Peters and I that jointly run the IDB thought that we needed to take a look at that. So at that point, there was essentially no data on this new virus because it was new. So and no information was coming out of China. Also, the studies were just starting. So we reasoned that in absence of any data about the SARS-CoV-2 virus, we could gain some information by what was known about other close relatives of this virus. And so it turns out that the old SARS, as Shane has referred to a while ago, is actually very closely related, looking at the structure as the genetic structure and so forth. And we looked back and we looked at what were the regions that were known to be recognized by the immune system back then with the old SARS. And we went looked in the structure of a new virus, and we found that those regions were very strongly conserved. So that meant that it was likely that this virus would be recognized and some of the same rules would apply. So with that in mind, we went ahead and started to make really comprehensive predictions of what could be recognized. And we then went back to the lab and started to synthesize these pieces. And next, we started experiments with non-exposed subjects. And why we were doing that? Well, one thing was out of necessity, because in late February, there were no cases to speak of in the United States or pretty much anywhere outside China. So we could not get infected samples to study. But we're also interested in a more basic, deeper question is, what happens the first time that someone that has never seen SARS-CoV-2 sees SARS-CoV-2? No? Shane was alluding to the fact that the development of memory is one of the key things in how the immune system reacts to a virus. And, you know, first impressions make sometimes very long-lasting memories. So we wanted to know, what is the first impression? What happens the first time around? And we started to develop these assays. Next. So as you probably detected from some heavy Italian accent, yes, I am from Rome. And at that point in early March, Europe was being ravaged by COVID. And there were many, many cases. And then we established collaborations with several Italian hospitals. And we started to have a process in place to get the samples shipped. And then at the same time, you have to deal with things that are very, very important. If you want to work with samples from people that have very serious infectious diseases, you need to do it safely. So you need to have the appropriate protocols in place, the appropriate approvals to handle this material, and also the appropriate protocols so that the people in the lab can do that safely. And that sounds like trivial, but it's not. And this is something you want to do correctly. You don't want to screw it up the first time and then fix it later. It has to be done correctly, always. At the same time, in mid-March, the bioinformatics study that we published, that we had performed, appeared in the press. And this was important because as I was mentioning, the function of the IDB is to share all this data. And so we put out this data and we basically made this data available to everybody in the world to catalyze and speed up the study of immune responses to the virus. Next. At this point, we in mid-March, unfortunately, we had samples. And through collaboration with various sites, but primarily UCSD, we were able to start to get samples from COVID convalescents. And we focused on COVID convalescents for the reasons that Shane was alluding to a minute ago, in the sense that we wanted to study cases of relatively mild, uncomplicated COVID disease initially as our first target. And why we wanted to do that? Because we wanted to study instances in which the immune system was able to take care of business by itself. So a success story, as someone that was infected, got rid of the virus, and didn't get too sick. And so we started to do these experiments, and next, at this point in time, also we started to send out our reagents, not only the information, but the physical pieces of the virus that we were synthesizing. We made it available all around the world, and we kept going, synthesizing more than 10,000 different viral pieces that people could study. Next, we continued to collect and to build a very large collection of blood samples, and we always could use more. We're looking constantly for volunteers. If you have had COVID, or you know someone that had COVID, go to our website. We love to have a blood donation to study. Why is it important to study a lot of people? Because COVID has a lot of faces. So there's people, as we were saying, that get very sick, people that don't get very sick. So why is that this variation? And also, everybody is different. Humans react very differently to viruses. The immune system is reacting very differently to each and different individual, and so it's very important. And by mid-May, we published the first landmark study in the scientific journal Cell. And in the next slides, I'll just give you the main take-home messages from that. So first of all, to our great satisfaction, our pieces worked and worked very well. And we actually keep hearing from all over the world that they work very well. Apart from the fact that we're happy that our stuff worked, the point that is important is that we could detect immune responses in 100% of convalescents. And this means that basically there is an immune response. There's been a tremendous amount of concern that the SARS-CoV-2 is not a good immunogen, that the immunity that is induced is weak and it's not good, and thereby a vaccine will be next to impossible to develop because it's just not a good immunogen. In the third and last bullet there, we can define quality of responses. So that is also a very, very important issue, because if you can't see and measure a response, then you don't know what's going on. And a very important thing is there a difference in immune response of the people that do well versus the people that don't do well? And if you can draw a picture, then you can discuss, this is a picture we like or we don't like, this is an immune response that is good, or this is an immune response that is no good. But if you can't generate a picture, there is no discussion to be had. Next point is that studying this average success, so it's nothing negative about being average in this case, this average success really establish a benchmark of what are successful immune responses. How do they look like? What is the balance of CD4 or CD8? Which piece of the virus are recognized? Good news, in the sense that this antiviral immunity matched expectations. The type of immune response we were seeing was similar to what we see in the case of other viruses, which are dealt with successfully by the immune system. Next slide goes through reiterate some of his same points. The fact that there was worry that this virus would not induce a proper immune response, but it does. And even a relatively mild infection causes a strong immune response, which is not transient. In fact, that is one of the things that we're doing right now. We're following people throughout time to see. Remember that graph that Shane showed you for smallpox? Obviously, we can't give you samples from 50 years ago because the virus is a few months old, but that's the idea, that we will follow through time and see how long the memory lasts. And also, it is still possible in some cases, the immune response might be overreacting, particularly in the cases that are very, very severe ICU. And we now have the tools to measure what's going on. Next. This has to do with what is recognized in the virus. Everybody's using the spike protein. In this graph is the S there. And the question is S a good target, apart from antibody responses? There is no question that S is a great target for antibody responses, but is it a good target also for CD4 on top or CD8 on the bottom? And it turns out that it is. It's kind of a complete piece, antigen. So using S in a vaccine will be able to bring to bear a response that is not only antibody response, but also a CD4 and a CD8. At the same time, if you just look at in this graph, there's many other pieces, many other genes that are in the virus, and you see that there is reactivity against several other pieces. So this can guide further vaccine optimization because it's possible to supplement or fortify, if you wish, a spike-based vaccine with some of the other components that also are recognized by CD4 and CD8 and then can bring additional firepower to your vaccine. Next. This was the most puzzling observation, and Shane and I and the rest of the team spent a lot of time looking at this data. So what you do in a scientific experiment, you do a negative control. So something that is supposed to be negative. So we also obviously had the COVID cases, but we also had unexposed people in the black. These were samples derived from 2015 that we have banked away. So there's no way these people could have possibly seen SARS-CoV-2. But it turned out that a good fraction of people that could not have been exposed to SARS-CoV-2 actually had this pre-existing reactivity, which was very, very peculiar. And this is one of the things that it's has been really generating a lot of interest. And the speculation is that it's in these blood samples collected in the 2015-2018, this reactivity is there because there is an immune memory, not against SARS-CoV-2, but against cousins of this virus that are the common cold coronavirus. There are 4 different main strains of common cold coronavirus, which is circulate broadly in humans, and they cause, in most cases, very mild infections and symptoms, and they are the common cold. And so this has huge potential implications, and I'm going to pass it on to Shane now for what's going on and what's next.
Yeah, thanks, Alex. So the scientific and media press response to that work that Alex just summarized was phenomenal. I mean, the paper was first published, I think, about 50 days ago now, and there are basically still news articles coming out about it every day, and we get more correspondence about it every day. Really, the response has been phenomenal, and in the scientific community, it's been hugely influential. You know, it's been really important that people know for vaccine studies and for studies in hospitals these basic features of what the immune response to COVID-19 looks like. And it's also been important for vaccine trials actually to know that some unexposed people already have some signal that people running vaccine trials weren't looking for before, and that might affect the results. All of this was done at LJI with patients from San Diego, and we were the first in the world to figure out these things. I want to say, you know, we succeeded, not because we're, you know, as big as some of the biggest universities, and we definitely didn't succeed because we had the first patients. We were first because LGI is really a special place with special people, and we're really good at understanding the immune system, and we've been working nonstop, that's for sure. But also really bringing to bear the incredible expertise of Alex Sette. I mean, he's like the #1 person in the world at identifying T-cell epitopes, and our other expertise for understanding what's important to measure about immune responses. And so we're continuing to now apply that to understand what is going on in these unexposed people who have some measurable T cells. And we're also spending a lot of time trying to understand hospitalized patients now. What's the immune response look like in people who have ended up with severe disease? One possibility is that those people are failing to make a good immune response, and that's why they're getting so sick. And the reverse possibility is also there, that they are making some sort of overactive immune response. And then there's the third possibility, that it's a different immune response that they're making. Again, as I said at the beginning, the immune response is complicated, and so we really have to make direct measurements to figure it out. To briefly comment on the common cold coronaviruses, so there are 4 of them. And they actually all 4 circulate in the US with different patterns. And so there are lots of questions here about are these T cells in unexposed people, are they actually specific for common cold coronaviruses? And if you do happen to have a recent exposure to common cold coronavirus and have a bunch of these T cells, does that actually affect your immunity to COVID-19 or not? And does it affect an immune response to a vaccine in a vaccine trial or not? And those are important unknowns. But if they did matter, right, it could make a big difference. And so we're working hard to make those measurements. And I mentioned this one already on the previous slide, and Alex has pointed out, right, that we're very open with all of this work. We send our materials and tools to basically anybody who wants them anywhere in the world freely to really try and accelerate international research. And I think we talked about several of the implications for vaccine development already, and so I'll go ahead and just jump to the team. This is the group here at LJI who's been working so hard on this. They're phenomenal people. As I said, I mean, we're first in the world to figure this out. It's because of the people we have in the place we're at, and that actually some of our important work is definitely donor supported. So if you're interested in helping out LJI fund this type of work, definitely let us know. And I will stop there, and we're gonna open it up to questions, because we're sure you've got questions, and we're happy to do our best to try and answer them in understandable language.
Thank you very much, Dr. Sette and Dr. Crotty. And now it's your opportunity to type in some questions. Use the Q&A at the bottom of your screen to type in your question. We already have questions coming in, so I'm just going to start off and read them where they're coming in. Not sure which one of you wants to take it, but here's the first one. Is a pathogen the same thing as a virus?
Yes, basically, you know, infectious diseases are viruses, bacteria, and parasites. And obviously it's viruses right now that are on everybody's mind because COVID-19 is caused by a virus.
Okay, here's another one. Is a 10-year cancer survivor who underwent chemo 10 years ago deemed to have a compromised immune system?
I think neither of us are qualified to actually answer that one. I have clinicians.
Can I take that one then? No, I'm just kidding.
Yeah, go for it.
I'm just
I mean,
kidding.
definitely if you had chemo within the past year, you're definitely immunocompromised, because chemotherapy preferentially affects cells that turn over a lot, and cells of your immune system fall into that category. But at a year, I don't know. That's definitely a question for
Right,
you.
but the question, I believe, was 10 years ago.
Yeah,
Right.
10 years
Yeah,
ago.
yeah.
Yeah, so 10 years ago, I think, again, I don't think we're qualified to answer the question, but over time, the immune system does regenerate to a certain extent. So the process by which memory is generated by a special pool of cells that are called naive cells. And those are the naive cells that can be taught new things. And that regenerates over time. So while I wouldn't say that I know that it's not immunocompromised, I would be optimistic that 10 years after the fact, that person would be able to respond perfectly well to new stimulations.
Okay. Here's the next one. What is the mutation rate of COVID viruses compared to other viruses?
My PhD is in virology, so I can attack that one. And I actually studied mutation rates. So basically, it's pretty much the same as other viruses, and they're basically two different things. There's the rate at which mutations occur, and then there's the rate at which mutations actually change something meaningful. So actually, most viruses mutate a lot. But they can't actually change very much. So year to year, they're actually the same virus, even though they were mutating at the same time. There's definitely recently been a major mutation in SARS-CoV-2 that swept the globe. That does look like it's a more infectious virus, and that's not surprising since it's newly entered the human population. That there's a lot of evolutionary pressure for on different viral strains. But so far, besides that, there have not been any concerning mutations that people have found.
Okay. In early March, we started to have samples in San Diego available. Were you able to access those samples?
Yes. So as I was mentioning, the relatively soon after virus arrives in San Diego, through collaborations with UCSD, we were able to obtain some of these samples, and that has been going on ever since. And initially, there was also, as I briefly mentioned, there was a little bit of uncertainty on how the samples should be handled. There are different levels of safety precautions that you have to use in the lab, depending on what type of samples it is. And it turns out that we work mostly with blood draws. And SARS-CoV-2 is not in the blood. So that to a certain degree, you can work with blood from infected people with still with a biosafety precaution, but not as much. But it took a while to figure out that we were going back and forth with different bodies, with the CDC, and so yeah. But the collaborators have been fantastic, and also as we figure things out, and we were able to accept blood from the outside, from people that volunteer for our studies. That has been a terrific thing. Again, in the start was a little bit logistically difficult because the institute was shut down and we were not allowing anybody from the outside to come in. We were kind of walled in and doing our research. But in order to accept someone from the outside to give a blood donation, we had to sterilize everything. So the clinical group really did a terrific job also to ensure that we could get blood donations from patients safely for everybody.
Very good. How are
Yeah, and I think I'll just add to that, since Alex was talking about, we're definitely still currently taking volunteers. We're interested in getting more blood samples from people, and in terms of why we're doing that and who we're interested in, there are some groups of people that it's actually very hard to study their immune response because we just don't come in contact with them. So like one of those categories is people who are sick right now, but not hospitalized, right? So if you've got COVID-19 today, but not severe enough to be hospitalized, we're very interested in a blood sample from you and what your immune response is doing, because that's quite informative. And then conversely, people who were hospitalized in the past but are no longer hospitalized, we're quite interested in looking at those people. And those are categories that we just don't have enough samples of to tell what's going on.
How are these viruses created?
I always tell people the whole reason you have an immune system is because viruses have a reproduction cycle of a couple of hours, right? And people have a reproduction cycle of 20 years. And so a virus is evolving, 10,000 times faster than you. So the only way for you to manage to stay competitive in that scenario is to have an immune system to protect you. So highly improbable events occur in viral evolution because there's so many events going on so fast. And viruses are essentially, coronaviruses have substantial recombination between different coronaviruses, and every once in a while those pieces stitch together in a way that can successfully infect a new species.
And in general, also, viral evolution and the interplay with humans has been facilitated by the fact that things that we did not used to get in touch with, now we get in touch with because of deforestation, because of changes in the environment. And also, it's always a numbers game. The population is greater, and the population density is greater, so that it increases the odds of just a statistical event happening. And obviously, as if a virus replicates in most conditions, has more of a likelihood of something like that to evolve.
How much help or hindrance is the CDC these days?
That's not really a question for us. I mean, it's a fair question, but it's not one that's actually directly relates to us. So for example, on the research side, it's actually the National Institutes of Health that funds biomedical research and provides sort of guidance to us, or us directly working with hospitals. And so people like Tony Fauci is the head of the part of the NIH that's infectious disease, and he's obviously been extraordinarily helpful.
Right. And it's sort of, so as Shane was saying, we don't interact as much with the CDC because most of it, biomedical research arena that we operate in is funded by NIH. We do have interactions. For example, if you need to import samples from overseas and things like that, you need CDC permits and things like that. And certainly they have been quite cooperative and certainly have not been a hindrance in those interactions with us for our lab.
Would increased antibody testing provide valuable information in immunology and vaccine development?
It would, and there are some huge sero surveys. That's what serology is, the study of antibody responses in the blood. So they're called sero surveys, where people do antibody tests of large populations of people. But you have to design those correctly, and you have to do them in large populations. And we expect to see more of them published. I mean, to me, the best one so far has been the one that New York State did a month ago, where they saw quite high percentages of people who had been infected already. And that's really what the sero surveys are useful for, to get a sense of how widely a virus has already spread in the population. For vaccine development, not so much. For vaccine development, really the studies that we were just describing are much more informative about immune responses. Is that fair, Alex?
Yeah, I think so.
How is your company financed? How is LGI? How do you bring in your money? Is it strictly research grants, or do you actually?
Right. So we're not a company. We are a research institute. So we are nonprofit. So our goal is to create and disseminate knowledge, if you wish, which is a good thing. I like that a lot. Of course, to create and disseminate knowledge takes money. And so we have grants and contracts from various sources. The NIH is one of the, by far, the major donor. We have also support from the Gates Foundation to the Michael J. Fox Foundation, from some revenue from licensing. If we discover something and it turns out to be useful and we license it to a company that wants to develop it, then the institute gets a little bit of a royalty payment, but that is not major. And then an increasing source has been to get donations from different donors, small or large. One thing that we do not have, we're not Harvard, we're not MIT or Stanford, we don't have a huge endowment that we can dip into buy the next fancy instrument. So but we do well. I don't know, Shane, do you want to add anything to that?
Yeah, I think the two specific things to add are we're a nonprofit research institute. The research that we just described was really expensive, and actually we had to start it without funding for it. We had to just essentially, hope that somebody would end up paying us back for the money we were expending, because we knew these were really important problems, and that definitely some of this work has been directly funded by individual donors who contributed to LJI. I mean, it's a real source for us, and an important one.
And important for us too. Here's our last question before we wrap up. How will we know when a vaccine candidate is good enough and safe enough to be rolled out?
Yeah, so that's a great one. Alex, you want me to start, or you?
Yes, go ahead.
So safety is always the #1 priority with any vaccine development, because vaccines are given to healthy individuals, and that's always been the case with vaccine development. And so they're really very rigorous safety precautions. And in fact, the whole first clinical trial done with any vaccine is the only input is safety. But then you can also collect immunological data on top of that. And so all the data that's been released so far has all been from phase 1 trials, which are very small scale, first attempts, and they're all focused on one single antigen of SARS-CoV-2, which is the spike. And what scientists are looking at are basically of that data that comes out, how good, both numerically and qualitatively, are the antibody responses that are being measured in those vaccine volunteers, and then how does that compare to an actual measurement of protective immunity. And so far there is evidence of protective immunity from our data, from the paper that Alex and I published about what a good immune response looks like. There's evidence of protective immunity data from antibody studies that people are doing on large group of people. And then there are data on protective immunity from monkey studies. So monkeys, you've really got to have an animal model of an animal that can be infected by the virus that you're concerned about, that gets disease kind of like what you see in humans. And then can you protect that animal with a vaccine? And so there's been lots of intense effort trying those experiments in monkeys and then looking at the results. And so far, several of the vaccine candidates have been able to protect monkeys from disease. And for several of them, it looks like it's a mix of antibody and T-cell responses. And so then it's a question of, can you get that much of a response in vaccinated people? And can you get those responses to be durable, to have immunological memory?
Okay, well, what a fascinating presentation. I want to thank both Dr. Alex Sette and Dr. Shane Crotty for a very informative presentation, especially in what we're going through today as the whole world. So I want to thank you all. I also want to thank the La Jolla Institute and for really giving us a chance. This was an exclusive presentation. This isn't something that's rolled out to everyone. So we want to thank the La Jolla Institute, and of course, they're represented by Claire Peister. Thank you, Claire, for coordinating with our team. I also want to take a moment and thank our events team, Valerie Bradley, who coordinated this, along with Lauren Crowley, Carla Connor, and the manager of our events, Claudine Casillas. Thank you all very much for all your great work. And thanks again to all of you as supporters of KPBS. As Producers Club members, we really appreciate it. And keep looking and watching for our Producers Club, our PC posts. We will be letting you know about our next events. So on behalf of all of us, thank you all very much and have a great rest of the day. And thank you again, Dr. Sette and Dr. Crowley.